Dear students, today's lecture is about the stress. Yes, the name of the stress state is generally the stress. The triggering factors are the stressors. If we want to emphasize the negative character of stress, we call it distress. The physiologically beneficial type of the stress is called EU stress, after the Greek word EU means true. The study plan can be classified in four big topics about the stress generally, the sources of the stress, the social stress, trials of Calhoun, and the pheromones. But you can see the details. Here on these pictures, I wanted to underline that not is the good situation like the human meditating at the left side. For the animals, the life is very, very stressful. The intensive keeping systems, the laboratory animal science, and even for the companion animals and the zoo animals too. So the basics of the stress is the general adaptation syndrome described first by Hans Scheyer, a Hungarian scientist working in Canada. The general adaptation syndrome consists of the classical trio, the suprarenal glands, the thymus and the lymph nodes, and the gastric mucosa. The increasing stress has a long-lasting influence on the health state, and the body can counterbalance it, but at what level it will be interesting, and what price. And I show you a proposed background literature, Robert Sapolsky, why don't zebras ulcer? Why don't get zebras ulcer? This is an excellent book about animal and human stress. So going into the details, I've taken the pictures from the original publication of Scheyer, 1965, published in Hungary in a book of the Academy of Sciences. The general adaptation syndrome involves both the center and the periphery of the nervous system and the endocrine system. You can see that the stressor and ACTH release, and after in the suprarenal gland you have influence on the white blood cells, and also in the gastric and duodenal mucosa. Yes, so this is the picture of Scheyer, and left side you can see the picture of the organs of rats. Left side you can see the control rats. First and the second row is second column is the rat after a long-lasting stress period. The first row are the suprarenal glands. You can see an increase owing to the stress. The second row is a thymus. You can see atrophy. The third line are the lymph nodes and the spleen. You can see atrophy too as lymphoid organs. And the last row is a gastric mucosa with ulcer. During the long-lasting stress, the organism is mostly under cortisol or corticosterone influence. In case of the human, cortisol or hydroxycortisone, and in case of the rodents and birds, we have the corticosterone. Both are glucocorticoids. And you can see that they have a highly importance in four topics, four fields. First is the intermediary metabolism. They will make everything to increase the blood glucose. They made lipolysis, mobilization of the fat, even muscle proteolysis, and in the liver there is a gluconeogenesis, making glucose from protein and amino acid. So this is the for increasing the energy, the available energy, the glucose in the blood. The second, in the nervous system, the glucocorticoids are regulating the ACTH and corticotropic releasing factor release and production. They contribute to the survival of neurons, especially in the limbic system. Too much is bad, too much will kill the neurons. And the maintenance of brain integrity. The field in the field of the reproduction, there is a signal for parturition by a peak of the ACTH because the fetus has no place. This is all begins with the stress of the fetus. In mice, milk production also requires the presence of glucocorticoids, and they have a permissive effect in the catecholamine synthesis and uptake of the cells. It means that the adrenaline and noradrenaline synthesis and uptake. And all of the essence of those are the survival during the stress. Today we have an integrated picture about the immunity, brain, neurons, hormones, and even cytokines produced by the white blood cells and other tissues like interleukin 1 and 6 and tumor necrosis factor. You can see the macrophage and interleukin influence, and you can see also that T and B lymphocytes are communicating by ACTH production, and also you see that through the nervous system, the rage and the anxiety has also a similar effect on the organism, like the long-lasting inflammation, right bottom, and the stress reaction. So all of those has a acute phase with the noradrenaline and adrenaline in the blood peak, and after the cortisol and corticosterone. And here you see the picture about the vicious cycle of stress. This is a feedback loop, and we call it in a medicinal language circulus vitiosus. It means that you cannot jump out. It is very difficult. This is a returning and returning. What is on arrow 1? This is a threat. What is a threat? Threat or stressor is everything which can knock the or push the balance of the homeostatic control, of the homeostasis. And it will send a message to the amygdala, arrow 2. Amygdala is a center of the fear and aggression, and not only the behavior but also the endocrine, the neuroendocrine regulation. But beside the cortex signal, the number 3, there is a preconscious signal to amygdala from the hippocampus. This is very important. It was proved that even in human, uh, especially in women, after a rope, after a traumatic event, they put the voice of the aggressor at the background, and without knowing why, her blood pressure increased and the heartbeat accelerated. So this is the unconscious part, preconscious signal to the amygdala from the hippocampus. And after you see that the amygdala, this is number 4, will produce corticotrope releasing hormone to the brain stem, and in the brain stem, arrow 5, there is an activation of the sympathetic nervous system. All the basic centers of the vegetative regulation is in the brain stem, like the blood pressure and the heartbeat. So this is increasing. This is the first part of the stress, and also via the spinal cord. This is the arrow 5. There is an activation of the adrenal gland in the marrow to produce adrenaline, and in the cortex cortisol. And in this phase, during the influence of the adrenaline, this is the fight or flight status, the acute status. But at the same time, if it's long lasting or chronic, the arrow 7 shows us that the cortisol, the circulating cortisol in the blood, or corticosterone, gives a feedback to the locus coeruleus. It is in the brain, and the name means that blue seed, blue nucleus, which in turn sends a neurotransmitter, in this case noradrenaline, to the amygdala to start the first, the second, third step. So this is a returning step. Here you can see on this picture the whole process, the whole feedback loop in details, summarized in details. And we have two strategies coping the stress. During the acute phase, or Cannon's alarm reaction, we have an organism under the effect of noradrenaline and adrenaline, and this is the fight or flight reaction. But during the chronic phase, during the effect of cortisol and corticosterone, there is another type of reaction. This is the tend and befriend reaction. And this is also called as bonobo strategy, because the fight or flight is not appropriate for gestant and suckling females. Shelley Taylor and her co-workers described in 2000 this tend and befriend reaction. The basic is that the oxytocin release in the brain, and the female reproductive steroid hormones and also endogenous opiate peptides are released. And this will produce a behavior, a tending behavior, protect, feed, care of offspring, and something which will facilitate it in a social environment by befriending, affiliating with social group to reduce the risk. And this is very typical for the bonobos. Bonobos are apes, are sub genre of chimps, and they are very social. They are using petting without aggressive behavior. So generally, this bonobo strategy, which is general in rodents, in laboratory rodents too, more frequent in females than in males. And now we have to see the curve and the effect of increasing or simply long-lasting stress and performance, or performance may be the biological answer. You can imagine a small cage for laboratory animals, or high population density, or a high ammonia concentration in the air, or simply if you are a student, you can imagine the approaching examination period. So you can see that in the resting phase, the performance is low. With increasing stress, with moving in time, you can achieve, say, the first successful exam, but after you get fatigue, and later on by the increasing stress, exhaustion, and because of the atrophy of the lymphoid tissues and the impaired immune system, immune answer, you will get very easily illness. It's very typical if you remember that many people get sick at the end of the examination period, close to the end. And of course, that we have to avoid a collapse and a death, but unfortunately, in animals, it is sometimes the case. And what happens during this long-lasting chronic stress? There is a balanced stress when the one elephant is the stressor and the stress, and the counterbalancing elephant is the organism with the physiological mechanisms. And by the endocrine changes, by the metabolic changes we described before, and the higher blood pressure and higher heartbeat, we can, and the animal can counterbalance the stress. But after finishing the stress, one elephant will eliminate it, and itself, the physiological functions are pathological. So what you can do in this case, for example, at the end of the examination period, you can change first to another beneficial stressful activity like physical activities. What are the main stressors, the triggerings in case of the animals? The first picture you can see the thirst and the hunger. If the chicken don't receive food and water, their vocalization will be higher and higher, or the heat, or the parasites in the body. Here you can see a giardia to prepare our next lecture about the pathology, because this is important in laboratory animals. You can see a gut mucosa and the parasitic giardia. And here you can see the list of the potential stressors. As I've mentioned, the hunger and thirst, climate, microclimate, parasites, and we will see next on the next pictures the place in the hierarchy, seeing in a zoo the apes, the lack of the partner, especially during the mating season or the estrus, lack of care or presence of predator, or itself the intensive farming system, depression in animals left at home, and lack of space and time for relaxing. See the experiments of Calhoun, lack of space and time for relaxing. Here you can see an illustration of the hierarchy in case of the apes, or guinea pigs mating, or caring of the maternal care. And now we have to see those sources of discomfort. And the unsocial environment can be divided in the room into the macro environment, this is outside the cage, and the micro environment inside the cage. Outside the cage, the temperature and the air humidity are important. You remember that for a mouse, we need temperature of 24, 26 degrees Celsius, plus, of course, the nest. Relative air humidity at about 50-60%. You need a ventilation in the individually ventilated cages, 16 per hour is the change of the air. Very, very low ammonia concentration is allowed. Light, dark periods, the basic is 12 to 12 hours, but it can be changed to 8 to 16. Noises are very important. Normal, so-called normal noises in the animal facilities are at about 50 decibel. So in this case, a classical music like Mozart as a background music may be helpful. Scents are very important. The pheromones of foreign species or own species but another sexes. What are the micro-environmental constituents inside the cage? The cage has a form, material, and measures. The measures are prescribed according to the EU regulations. And installation, bedding, and environmental enrichment, which is today legally obligatory. Concerning the environmental enrichment, we can use running wheel, for example, and we have to mention the pre-MEC principles, or in a slang, scientific slang, this is the grandparents' law. This is help without interest, or making something without a treat, without a recompensation by outer sources. There is a congenital driving force to act. This is a driving force, which is self-reinforcing and self-rewarding. See the running wheel. The animal can release dopamine and endogenous opioid themselves. We don't have to give a treat, extra feed, for example. And here you see that this legally imposed environmental enrichment, which is, for example, in case of a human, a Hundertwasser house in Wien, but to the animals, the possibilities of the enrichment in the social environment is the group keeping, a very well educated animal caretaker and researcher. The feed type can be also like that, you have heard about it, the distribution on the bedding, and the sensory stimuli, like the acoustic stimuli, a continuous classical disco like Mozart, smells, pheromones, with or without, hiding possibility, physical space, nest material, toy like chewing treat, and in the next some dias you will see some pictures. In a modern cage block, it is not a environmental enrichment outside. This is the individually ventilated cages, but inside you can do it. For beagles, you have to use this training equipment to jump and hide, etc. And you can use tubes and different hiding possibilities, this so-called igloo, or even half of apple as a environmental enrichment. But never mind, the big companies are producing environmental enrichment tools. You can see here the different tools of the data set, but many can be found in the website. But be careful, the too successful enrichment is dangerous. For example, you can see that here in the pet shop, a snake is moving the running wheel, so this is too much. And a very important field of the stress in the field of the laboratory animals is the social stress. The social stress was scientifically described first by Christian, who studied the seasonal changes in the weight of the adrenal glands in woodchuck or marmota, and he has found that during summertime, this is the time for meeting and mating and fighting. There is a larger adrenal gland in the cadavers than during the quiet winter season or winter sleep, of course. And here you can see the original diagram of the author about the uh, uh, regulation of the population density. But you can, of course, see the details in this diagram. It's very useful. But I made it more digestible. And here you can see on this brain map. In the left with red, pressure from social stress and will influence the population density. Right side you can see the most important factors. The anterior pituitary gland, the growth hormone production will decrease and the ACTH will increase. The adrenal gland, the medulla cortex, will produce the cortisol, the first the adrenaline and after the cortisol, and the gonadotropin in the anterior pituitary gland, the FSH and the LH will decrease. If there is a lack of feed, the case is more difficult, more severe, but this is not a sine qua non, not a prerequisite of the social stress. It is an extra factor. Owing to the decreased gonadotropin, more abortion is found and abnormal maternal behavior. and of course that the reproductive functions in both sexes decreases, and because of the compromised immune response, the mortality increases, and less reproduction, more mortality may make a balance in the population density. How to study the effect of crowdedness on the social behavior of animals as a model for humans. These are the Calhoun's classical trials. The criteria of choosing the animal model, the rat, will be shown in the next picture. The next slide. The experimental design was a deprivation of feed, drinking water, or and hiding place, but in every case there was released a overcrowdedness, a very high population density. He made three trials. Trial one was an open field trial. Trial three was a rat city. Both are very interesting, but I will give the details of the second experiment. But returning to the decision matrix of Calhoun, we have told about it during the animal models. You can see that the characteristics of desirable in animals used for developmental population ecology. This is the original picture of the original publication of Calhoun in 1954 in the Journal of Academy of Sciences in New York. Left side, there is a list of the characteristics like biotic potential, diurnality, handling, genetic strains, food dependency, etc. And on a line, row 1, you can see the possible species, Norway, rat, mouse, etc. And 4 crosses plus means that very, very good for these purposes. And 0, there is no not able to make in the experiment. Only I give you one example. Chimpanzee would be the best model for human, but you see the genetic strain 0 score. It means that no genetic strains in case of the apes, so bad score. Finally, as a summarized total, the Norway rat received 42, and this is the best model for population density. Here you see the original experimental spot of Calhoun in his own store, own bar, and you see that he divided into 4 boxes, and from one boxes to another there is no gate, but there is a balcony above. We cannot see it, and through the balcony and this plateau and the staircase, one by one, you can go from one box to another. But of course, that one big male standing on the platform can prevent this change of your box. So what happened? At the beginning, Calhoun placed 10 female and 10 male in each boxes. This was just very nice as a population density. But in 3 weeks, and of course I've told you that applied food hoppers and water fountains increased the tension because they distributed the food and water only slowly. So the animal had to make a row to receive the food and water. But what is important, in 3 weeks, the population density changed. change of the territorial distribution of the individuals. On the left side, you can see my own picture that in box 1 and 4, there was a first and second biggest male making a harem with 10, 13 females. And all the remaining males and, of course, the less females were distributed in the box 2 and 3, causing a high population density. And the population density and the chronic stress and the compromised immune system and the lack of maternal care caused high mortality. You can see right side that in the middle, box 2 and 3, high mortality rate. Especially in case of the pups, the abnormal maternal behavior, you can see in this picture that no nest building, no suckling, and even no control of the pups caused a high pups mortality. And this is the summary of Edward Hall, The Hidden Dimension. This is an excellent book about the behavior of humans and animals, published by the Anchor Books, New York, 1969. Even the rat, hardy as he is, cannot tolerate disorder and death like man. He needs some time alone. If pregnant rats are harassed too much, they have increased difficulty in bringing pregnancy in term. Everything in box 2 and 3. The sex modes of the rats in the sync behavior is the disrupted behavior. Was disrupted, and pansexuality and sadism were endemic. Rearing of the young almost totally disorganized. Social behavior of the males deteriorated, so that tail biting broke out. The social hierarchies were unstable. So this is the long-lasting social stress. How can you see by one glance that there is a problem with the social behavior? You can see the action catalog or ethogram. You can compare the to the normal orientation, comfort movements, metabolism related. Yes, 5-10 minutes. Metabolism related, attack or protection, territorial, race preservation, nest building, social and other behavior, the actual to the normal, to the characteristic to the species. The more social an animal is, the more complex is the ethogram. So you can see that for the guppies, you have 5 different types of action catalog. For the golden hamsters, you have 9. And for the mouse, we have 18 with very, very different grooming and other behaviors. And what is the medium of the social stress? Because we have seen that not directly the lack of feed and not directly the physical lack of place caused this problem. And these are the pheromones. The pheromones or the science of pheromones or science of exocrinology was discovered in the group of the insects. You see here grasshoppers, and ladybirds, the social life and sexual life are organized by the pheromones, the chemical compounds called pheromones. For two dogs, the nose contact is more efficient than our identity card. So the pheromones, these chemical molecules, are perceived by the vomeronasal organ. You can see in the middle of this picture, below the main olfactory epithelium. And the classification of the pheromone compounds are releaser, like the bombitol of the mite, to attracting the male. 2-3 molecules can attract the male from 2 kilometers. Primers are which can induce the cycle, like the Witten effect. Signaling are, for example, for the recognition of the offspring, ewe and lamb, and doe and pups. Here you can see that a doe just marking her pups by the mandibular gland. And modulator type of pheromones are for the synchronization of the cycle. Some males which are living in a distance from the females are developed a flamen behavior. They open their mouths to facilitate the perceiving of the pheromones, the achieving the pheromones to the vomeronasal organ. In the rodents, the pheromones has a very, very important reproductive functions. I summarized it because you have heard about during the zoology lecture from Professor András Berseny. The Vandenberg effect is a stimulation of the sexual maturation of growing females by the pheromones of an adult male's urine. The Lee-Boot effect means that without males, the group of the females will get in anestrus. The Witten effect is the second phase. This is a synchronized estrus in the anestrus female group after entering a male or the scent of a male. And the Bruce-Barton effect means that after fertilization, 1 maximum 2 days, no implantation occurs if another more aggressive male pheromone, will arrive to the female. Aphrodisiac, some books call this as a abortion, but this is not scientific. Aphrodisiac is produced by the vaginal mucous membrane of the golden hamster, and this is an invitation for mating because the male is not accepted. The hamsters are very solitary animal. Only during the estrus time, every 4-5 days. And in human, what is the situation? First, the vomeronasal organ and its genes are known. Publication in the Nature, a Nobel Prize. The fetus has vomeronasal organ. In the adult, 50% atrophied. The androstenone through a gene can cause GnRH LH changes in women. and the androstenol, both are part of the male pheromones, is a GABA A receptor modulator. Grammer stated that the human sense of smell, both chemical and pheromones, has by far been underestimated. This is the situation in the human. In the human, the chemosensory communication is proved because there is a synchronization of the cycle amongst women, and we have also a nursing mother suckling baby recognition by the scent of the mother's skin and milk smell. And people has a smell preference of another. We know also that the vomeronasal genes and the major histocompatibility genes are expressed from the same neurons. This is the reason that we don't like the sexually, we have no attraction in the scent of the relatives, and some people told that the love at the first sight is rather love at the first smell. And we have also a human has a sexual dimorphism in this field. The women are more sensitive to the smells. So we can solve the problem by the evolutionary hypothesis that the effect of social life during the human evolution, the big families and living in groups, uh, was not an advantage, that everybody knew that a female is in estrus. So human became more visual oriented. The speech for information changes developed the speech. There was an atrophy in the vomeronasal organ in the majority of the adults. Decreases number of smell receptor genes, and they became pseudogenes, and the genes of odorant binding proteins extincted in the human, like in the orangutan, and the gene of odorant binding protein doubled in other species, in the rodents, horse, and cattle, where this is highly important in the communication. Finally, we have to see the actual veterinary application. We use the boar spray with delta for androsten to synchronize the virgin jilts. We can use the canine feline facial fractions, the Selgian, Feliway, and Felifriend, for relaxant and antidepressant and change of feet. And we have also the dog appeasing pheromone, I will just show you, which uh, consists of two social odor pheromones, the mentioned facial fractions and the appeasing pheromones from the gland, Sabates gland of the milk producing gland, and this is the two is in the dog appeasing pheromone, which is used in case of the separation related problem, in firework fears, puppy settling, car travel problems, kennel barking, and in clinic anxiety cases, it proved that this is very advantageous. This is not a hormone, and no side effect like the medicine. Some minutes later, but we had to close this topic, because the next time I will speak about the pathology of the laboratory animals. So once more, thanks for your attention and this small extra time.